to try to start to put the foundations in place so that we could move forward together in a directed fashion, focusing on areas of research importance that would have been recognized jointly by Africa and Europe. And it builds upon an agreement between the African Union and the European Union, who are going to launch lots of calls for research initiatives that will be Africa-facing. So we thought by having these calls in place, it would give us a head start as to so that our alliances were already organized, our research questions have been identified, and that when the appropriate call came about, that we'd be in a position to go forward. Could I have the next slide, please, Andrea? Thank you. So we formed 20 of these clusters, and they were launched 2 years ago now. And there are 9 in public health, of which ours, the Advanced Infectious Disease Research and Training, is one of those. And so we want to build around that, and we recognize that one of the key drawbacks for proper scientific progress is to have all the fundamentals that one would need to pursue high technology research, both in Europe and in Africa. And that's a result of ability to and opportunities to be trained and carry out training, but also the actual hardware necessary to carry out the technology, and indeed in certain settings, the correct location, laboratory space that would be suitable to house the equipment necessary for these technologies. And it's also, as you can see, that there's the green transition, which means that by housing all of these technologies in the appropriate countries, then that reduces the need for expensive and carbon-consuming travel between the different laboratories, because we can actually carry these out, communicate as we're communicating today, but with all the infrastructure in place on the ground where it's needed. And that will then inevitably improve our capacities for science. Next slide, please. So this is the geographical distribution of the membership. You can see it's color-coded. So in dark blue, there are the co-leads. So from Africa, it's the University of Ghana and Gordon Andware at WACBIP. And it's ourselves in Glasgow and our colleagues here at the University of Tübingen who co-lead within Europe. We then have other partners who are core partners. In other words, their institutions are also members of either ARUA or the Guild. And one of the nice things about this is there is no restriction upon invitation for other institutions to become members. They can be invited by the existing membership to join in. So you can see in the teal color a number of different institutions that have been invited to come in because they offer crucial advantages to the goals of the cluster. Next slide, please. So where are our expertises, what we're going to offer training in, where we're going to carry out our research. So we have human-controlled infection and vaccine development with Tübingen, Radboud and Semel as key players in that area. The pathogen infection cell and molecular biology with Glasgow, Bern and Kamuzu University occupying expertise in that area, and then bioinformatics and genomics, which is University of Ghana, Makerere, and University of Cape Town. So there's very complementary. These, of course, just not complete divisions. There's a lot of bleed between these areas, and so we expect that this overall composite strength will allow us to put forward exciting proposals that we will believe will be competitive for any African-facing calls that come. Next slide, please. All right, so that's the overview. Now to the business. As Andrea said, we're going to have 2 talks each time. There will be roughly half an hour. I'll switch my screen off, but I will reappear with 5 minutes to go. So that you're reminded that you need to be thinking about wrapping up, because we'd like to have questions and time for those. And so the first speaker, it's actually a great pleasure to be able to introduce Taco to you all. Taco, a few years ago now, started out as a PhD student in Chris Janssens and my lab in Leiden. He did extremely well. He graduated cum laude, and I did threaten him that I have a lot of photographs that I could show at this point to embarrass him. He then reminded me that he's chairing in a couple of sessions' time where I will be speaking. So I thought probably it's better not to show any photographs whatsoever and simply allow Taco to talk. But it is a great pleasure to introduce him, and I'll pass over to Taco now to tell you his story.
Thank you, Andy, for the introduction, and thanks a lot to this great honor to be the first ever speaker for the CoRE-AID webinar series. Very delighted. Also very delighted that my daily board of the Works Council allowed me to actually skip on the meeting to come and present here. Yeah, I have control over the slides. That's cool. So today I will tell you not only about a parasite that is, of course, a devastating health problem in Africa and other subtropic countries, but also to try and convince you that it's an excellent model to study general mitochondrial biology. But let me first introduce our institute a little bit, because I think that's one of the core purposes of this core, is to foster collaboration. And in addition to a wealth of infectious disease research that's going on in our institute, which is way too broad and for me to present here, I just want to briefly highlight the malaria research that is happening here in the Radboudumc and the Radboud University in Nijmegen, which, as you can see, is slightly dominated by white males. So happy to make some change in that in the future. But what is very diverse is the topics that we are working on, which range all the way from really the epigenetics of malaria parasites by Richard Bartfai through parasite biology and mosquito behavior, all the way to controlled human infections, translational work in the form of transmission blocking vaccine studies, and studies in the field on epidemiology and particularly malaria transmission. So we really cover the research in malaria and the Plasmodium parasite all the way from the basic biology of the parasite to transmission in the field. My own team focuses on the molecular and cellular biology of the parasites, and currently there's still 4 PhDs and 1 postdoc that are either directly under my supervision or work in very close collaboration with my team. And I won't be able to talk about most of the projects, so I will quickly highlight the active projects. The first one is by Ezra Beckering, who's working in the group of Matthijs Jorre on the very well-known transmission blocking vaccine candidates PFS 4845 and PFS 230. In close collaboration with partners in Canada, he's been able to elucidate the structure of this important transmission blocking vaccine complex, showing in great detail the structure of the complex, and also how 230 is wedged in the 4845 via a very small extension of the protein. And by removing this little extension, association of 230 with 4845 falls apart. Laura Ackerman started to work on a project that has its origin already a couple of years back in a close collaboration with a local small spin-off company called Taco Kooij, working on finding the mechanisms of uptake and of action of the pentatenamides, a new class of antimalarials that are in clinical development right now. And we had a couple of transporters that we suspected might play a role in this. But analyzing these in further detail showed that all of these were actually apicoplast localized, making a 180-degree switch on her project. And she's now completely refocusing on transporters of the apicoplast, also in close collaboration with the Soldati lab in Geneva. Milou Stevens works in the group of Pascal Mise on dengue and Zika infections and how these infections have an influence and are influenced by mitochondrial metabolism of the mosquito. In addition to that, she's also started, and that might be of interest to some of the people in the cluster, in spatial transcriptomics of infected mosquitoes. But today I will talk about mitochondrial biology, and this project, the most recent results that I'm going to present today have been generated by Sylvia, a PhD candidate in my team, and Arena, a postdoc in the lab of Lara van Niftrik in the Radboud University, who really work as a team on this project where we try to pin down cristae formation in the malaria parasite. So mitochondria, as you all know, are a hallmark of eukaryotes. And I always like to start by asking to think about what actually makes a good eukaryotic model. So most of what we know about mitochondria has been studied in a very limited set of organisms, most of them in animals, such as model organisms like mouse or C. elegans, Drosophila, yeast, or plants. But the huge variety in eukaryotic life is largely ignored in these fundamental studies. And also the malaria parasites, of course, is quite different from all these standard model species. However, I think makes a really powerful model to learn even about those highly studied model organisms. And by the end of this talk, I hope to have convinced you why. The malaria parasite life cycle is actually part of that answer. As you all know, the malaria parasites transmit from the mosquito first to the liver, then to the fast asexual replication, which causes pathogenesis, to the sexual stages of the parasites. And it's this transition which is also reflected in the morphology and functioning of the mitochondria, which changes dramatically through stage transitions. Some other facts about the mitochondria of the parasite are that for a long time we thought that all life cycle stages are characterized by having just a single mitochondrion, as opposed to our own cells, which are often have tens, hundreds, or even thousands of mitochondria per cell. The mitochondrion is a validated drug target, particularly atovaquone, which targets cytochrome B, BC in the respiratory chain, is a very strong target for this potent antimalarial. Another interesting aspect is that the mitochondrion is often very tightly physically associated with the apicoplast. In these pictures, which are Plasmodium berghia parasites with a red mitochondrion and a green apicoplast, you can clearly see that these two organelles often are very closely together, although not in all cases, such as the sporozoites at the bottom. There's many fascinating peculiarities that set out mitochondria from the malaria parasites from their host mitochondria. And one of the things that has been recognized for a long time is that it has one of the most, one of the smallest mitochondrial genomes known, with coding only 3 genes, cytochrome B and COX-1 and COX-3, all part of the OXPHOS complexes. The malaria parasites replicate during their pathogenic stages in the blood through a process called sclerogeny, in which the parasites produces huge amounts of biomaterials to produce all their offspring cells in a single go. And it's been long questioned how the organelles that the parasite need for all these daughter cells are replicated. And this was a project that Julie Verhoeven, who is now working in the lab of Julian Reiner in Cambridge, set out to discover. And she used a combination of fluorescence microscopy, expansion microscopy, and reanalyzing a lot of FIPSEM data, so three-dimensional reconstructions of the malaria parasite, to build a nice picture of what is happening. And what you see here are the nuclei in light blue, the centriolar plaques in purple, which all attach to the apicoplast in yellow. And the mitochondria forms a, let me show the movie once again. It was supposed to be repeating. And as you can see, the apicoplast nicely follows the centriolar plaques. But the mitochondrion is somewhat away from this and also forms a big network. But both organelles are still intact and form one continuous lumen. When she analyzed a number of three-dimensional reconstruction of cells in different stages of development, she clearly identified how the apicoplast in yellow here at the bottom divides first prior to the mitochondrion, when the apicoplast is already segregated entirely here, while the mitochondrion is still fully intact before the mitochondrion starts to divide asynchronously, so breaking apart in first bigger clusters and slowly breaking up into all the single mitochondria, which are divided across all daughter cells. When moving on to the next stage of the parasite's development, moving to the mosquito, the male and female gametocytes still appear to have a single mitochondrion when observed via fluorescence microscopy. And when Cas Boshover was actually working on mitochondrial transport proteins, was analyzing some of his mutants during activation of the male and female gametes, he observed that the females still have this this ball of mitochondrial staining very close together and not distinguishable from each other. But in the male parasites, he noticed that some of the mitochondria started to round up and segregate. And he was wondering whether this was a random phenomenon from the mutant, or that this might actually have something to do with the general biology of the parasite. And with this in mind, we set out to really look in great detail at the three-dimensional structure of the gametocytes. And this was work done by Felix Evers, who recently graduated in my team, who made complete three-dimensional reconstructions using FIPSEM of different stages of development of the gametocytes, but in particular of the mature male and female gametocytes, which you see here. And the next animation shows the same colors as depicted here. So here in red, you see the red blood cell. Inside is a female gametocyte, which will slowly appear now. On both ends of the parasite, you see in light green the ER. In the middle, in teal, the nucleus and the osmophilic bodies in pink at the surface. And as you can see, the red mitochondrion has the yellow apicoplast wedged in between and is completely intertwined with some interesting and peculiar extensions of the nucleus. So the nucleus doesn't form the typical ball-like structure, but actually has extensions, which we don't know the function of yet. They're free of DNA, and they're there in both male and female parasites. And they're, as I mentioned, strongly entwined with mitochondrium and the apicoblast. When Felix looked a little bit more careful at the data, and this was only 2 days prior to submission, he suddenly realized that there was something that he had missed all along, and something that all scientists looking at mitochondria had missed thus far. And as you could tell from the fluorescent microscopy data, it was not obvious that, or it appeared that the mitochondria, just like in the asexual blood stages, form a dense network. But when he really re-examined the FibCEM data slice by slice, going through the entire depth of the images, he realized that some of these mitochondria were never actually fully connecting with each other. And in the top left, you see the red arrows pointing at positions where mitochondria come very close together, but the membranes are never actually fusing and the lumen is not continuous with each other. And when he followed this all the way through, he realized that in fact, these gametocytes have between 4 and 8 mitochondria per cell typically. And this is true for both male and female parasites, and already from very early on in development. And this is the long-standing dogma that malaria parasites have a single mitochondrion throughout their life cycle. Another striking observation, which was already known in literature but never really been paid much attention, was when comparing asexual blood stages with sexual blood stages. In the asexual blood stages, the mitochondria are roughly the same size as the gametocytes, but as you can see here, the lumen of the mitochondrion is entirely empty and there are no internal membranous structures. In the gametocytes, we see typical internal membranous structures of the cristae that are so characteristic of mitochondria in most eukaryotes. So malaria parasites present themselves as a fantastic model to study de novo cristae formation, where we have cristae present in liver stage parasites, as identified on very old EM pictures, who lose their cristae in the mitochondrion entirely during asexual blood stage replication, before building them again de novo while developing into sexual blood stages. Felix then also went on to look at the proteomes of these different stages of enriched mitochondrial fractions and confirmed a long-standing knowledge that malaria parasites during their rapid asexual growth largely rely on glycolysis, as represented by the protein abundance in black of mixed asexual blood stages. While in the gametocytes, represented in gray, pathways such as the TCA cycle become much more prominent. This was also reflected in the presence of OXPHOS complexes. So mitochondria are full of very complex multi-component protein complexes. Most well known, the OXPHOS complexes allowing the mitochondrial respiration, including ATP synthase for the ATP production. Of note, malaria parasites lack their complex 1 and have a single protein of bacterial origin to replace its function. And so a way to study this is a protocol called complexome profiling, whereby you separate protein complexes, for example, in our case, on blue native gel, before analyzing different fractions of different sizes via mass spectrometry. And in doing so, we identified a whole bunch of new components of the different OXPHOS complexes that co-migrated. So here on the horizontal line, you would have the different fractions of the blue native gel, and intensity shown in yellow or red of the amount of peptides identified. And as you can see here, we have a whole bunch of proteins co-migrating at exactly the same size, representing the complex 3, and in this case, complex 4 of the respiratory chain of the malaria parasites. And like for the single proteins, as you can easily see here, when comparing asexual and sexual blood stages, we have a massive overrepresentation of these complexes in the sexual stages compared to the asexual stages. And this was true for all 3 of the OXPHOS complexes. There is one other complex of the mitochondria, which we were very keen on identifying, and which is known as the MICOS, the mitochondrial cristae organization system, which sits at the neck of the cristae. So we here have the outer membrane of the mitochondria and the inner membrane, and we have those invaginations of the cristae where all the OXPHOS complexes are represented. And at the neck, we can find this complex, which in malaria parasites was inconspicuous, and we could never really identify any parts of this complex except for MIC60 in the genome of the malaria parasite. Other components were never found, but in the complexome data, we did identify one additional component called PF-MIC19. Here you see alpha-fold reconstruction of the Plasmodium sequences compared to the yeast sequences, which clearly show the conservation of all the important parts of the important domains of these proteins. So this is the project that Sylvia and Irina worked on very intensely. And we decided to target these 2 proteins and these 2 genes in the Plasmodium parasite in order to finally answer the question whether MICOS is responsible for cristae formation, or if they are just there for stabilization of the cristae. So here you see a Western blot of asexual versus sexual blood stage parasites, and you can clearly see that in asexual parasites, neither MIC60 nor MIC19 are present, where in gametocytes, the protein can be identified, even though the amount of protein appears to be very low. And this was probably also the reason why, no matter what we tried, we had an HA tagged to the protein. We could not identify it by standard fluorescent microscopy, nor expansion microscopy, which we hoped would free up the epitope for antibody binding. So here we have expanded gametocytes with mitotracker stained, and we can really nicely see the mitochondrium. You might even be able to identify some of the individual mitochondria there, but unfortunately we could not identify MIC90 or MIC60. We then went on to generate gene loss of function mutants, deleting the genes entirely, and we also managed to generate a double mutant lacking both MIC19 and MIC60. And parasites, to no surprise, grow normally in asexual blood stages, but also, very surprising, produce gametocytes in normal numbers, which also seem to be activating at normal rates. There was a bit of a reduction in oocyst formation after feeding, but most of the parasite or the parasite could still transmit to mosquitoes and form sporozoites. However, when focusing on the morphology of the mitochondria, we really clearly see a defect of the parasites. So whereas in normal parasites, most of the mitochondria would be tubular, or sometimes you would see a bit of these intermediate phenotypes, Sometimes when the parasites are not feeling all too well, we see these rounded up mitochondria. And this was something that was much more regularly observed in all the mutants. We use then a plugin for Fiji to really quantify all this mitochondrial morphology. And in all cases, we saw significant effects. In summary, the mitochondria appear to be a bit smaller, more spherical, and less branched than in wild-type parasites. And this was also the case when we zoomed in even more at an EM level. And for the sake of time, because I already see Andy, I'll skip on this one, but I'll move straight on to the next one, which is tomograms of the gametoside. And in the top left, you see the wild-type parasites NF54. In dark purple, it's the outer membrane. In light or in pink, the inner membrane of the mitochondria. And then in yellow and red, you see the cristae. And red cristae mean that these cristae have a clearly identifiable connection to the inner membrane of the mitochondrion. Whereas if they're yellow, this connection is not observed. And if you compare the wild type in the top left with the single mutants at the bottom and the double mutant at the top right, you clearly see that even though the mitochondria appear to be so relatively similarly shaped, the cristae with a connection to the inner membrane are far less abundant in the mutants compared to wild type. And we have a lot more cristae that appear to have no connection whatsoever to the inner membrane. So we managed to quantify this as well. Looking at
we do have to give Pocha a chance here. So if you could get to the end quite quickly, please. Sorry.
Yeah. This is the last bit. So if we quantify this, the cristae junctions, so where the parasite of the cristae connect to the inner membrane, are strongly reduced upon loss of the mycos complex components. So in conclusion, we have a refined model of organelle division and segregation of Plasmodium. We've identified that gametosides have multiple mitochondria and a bunch of other deviations, which I didn't have time to go into when comparing asexual and sexual blood stages. But most importantly, the malaria parasites provides an excellent model to study cristae formation, as we have this situation where cristae are completely absent in asexual blood stages. And as we now identified, the loss of mycos did not result in a loss of cristae, but just a loss of cristae junctions. And with that, I'd like to thank all the people that contributed to this work, but most importantly, the people in the team and the students and the people at the malaria unit. And I'd like to thank you for your attention, and happy to discuss anything or answer any questions you may have. Thank you.
Super. Thanks very much indeed, Taco. I take it the questions are supposed to be going in the chat. I've not seen any yet, so please type them in quickly. We really only have time for one or two questions. So I'll kick off. So Taco, is it your intention then could you take advantage of the inducible gametocytogenesis and then look at the timing of cristae development and see whether the timing is affected by any of the mutants, or look for new mutants that might influence that and their impact?
Yeah, you just revealed the grant submission we just submitted earlier this month. So particularly the early stages, because we have looked at some stages, because we have some contamination of earlier stages in the sample that we had. So we had down to an occasional gametoside stage 2, where we didn't see much of a difference in cristae formation. So we now want to work with the line from Matthias Marti, where we can isolate the early committed gametosides Gametocytes and early stage gametocytes to look at crystal formation in those parasites.
And do you think there might be any differences between same and second cycle, next cycle commitment?
Not sure about that. Never really thought about it too much. I do think that it's actually a very early on process. Right. I have the suspicion that we may be able to tell on the basis of mitochondrial morphology already well before we have many other markers available, whether it's going to be gametocyte or not.
Okay.
But that's gut feeling.
Right. Stefan, you're unmuted. Do you have a question?
Yes, thanks. I have a very stupid or naive question, Taco, concerning the mode of action of atovaquone. Maybe it's so totally obvious, but I don't quite understand. So if for oxidative phosphorylation isn't really required in asexual parasites because do glycolysis, so why targeting the BC1 complex would result in parasite death?
Yeah, so that's
stupid, naive question.
Yeah, so this is work by Aki of Idea, and he clearly demonstrated that it's the recycling of ubiquinone, which is the only real target for atovaquone activity.
Yeah.
Which is required for pyrimidine biosynthesis. So it's really for parasite replication then, and not mitochondrial functioning per se.
Yeah.
And Christine has a question in the chat. I think that's falciparum, isn't it?
Yeah. So exception of the very first pictures I showed, which was Bergia, but we do all our work in falciparum these days.
Okay, right. Fabulous work, Taco. I think we should draw it to a close there because let Pacho. Pocha has been waiting in the stables, eager to get going. So we'll switch over.
Some online applause. Thanks, Taco. Super.
No, it was excellent work. Thank you. Do we just do that? We do. Okay. So Pocha, Kamudumuli, Kamuduli, apologies, Pocha. PhD student at the pathology department at Kamuzu University. He's been working on fungal biology, and he's going to tell us more about his work. So I'll pass it over to you, Pocha.
Hello, everyone. Welcome to my presentation. So my name is Pocha Kamudumuli, a PhD student at CoRE-AID. And I really thank you for giving me this opportunity to present about my study. I'll be talking about fungal infections, but I'm going to focus much on histoplasm and aspergillus. So I can move the slides. Next slide. Thank you. So that is the outline of my presentation. I would like to give you just an overview of fungal infection. They really pose a risk regardless of region. They cause devastating infections, can be localized or systemic. And unfortunately, very few fungi are being diagnosed. That's the case with Malawi. We are very limited in terms of diagnosis, which leads to undiagnosed fungal infection, and definitely some are being neglected. So, sorry, I think the slides are moving quite fast. Giving you a summary of epidemiology, you can appreciate that fungal infections are causing devastating rife. Approximately 150 million people suffer from fungal infections. and actually people with underlying health problems and weakened immune systems such as HIV, cancer, chronic lung disease are at high risk. No wonder about 1.7 million deaths per year occur, many associated with HIV and AIDS. Because of the devastating effect caused by fungal infections, WHO for the first time has come up with fungal priority pathogens list, which is categorized into critical group, high group, and medium group. And that does not leave out the aspergillus, which falls under critical group, as well as histoplasma, which falls under high group. We can agree based on the devastating effect caused by these two fungi. They cause histoplasmosis and pageurosis. Of course, they are neglected. Unfortunately, their symptoms are not specific. They mimic TB. They can cause pulmonary or systemic disease. And they can present in form of fever, weight loss, edema, and lymphadenopathy. So based on that, WHO further came up with areas of priority to combat the listed fungal pathogens, focusing on 3 critical areas. That is public health intervention, surveillance, research, and development. So looking at one of the 3 critical areas, our study is trying to promote surveillance part of it. And when it comes to surveillance part of it, we saw that we are failing to improve in surveillance area just because of diagnostic challenges, which are affecting a lot of countries. And under diagnostic challenges, the main problem is unavailability of reliable diagnostic tools, and of course, fungal infection being hindered by other diseases such as TB, and of course, some are being neglected as such that they're not being diagnosed. So moving forward, we appreciate that we have data. Cases have been reported of histoplasmosis and aspergillosis in several studies. But when we look at the data, you find that most of the data is coming from developed countries as compared to Africa. Unfortunately, that's where cases of HIV and TB are high, giving a possibility of having undiagnosed diseases. We go back to the challenges of lack of diagnostic tools and use of poor diagnostic tools. We are talking about microscopy, which is largely being used in most of the developing countries, which lacks sensitivity and specificity. And in some few countries, there will be culture, which is laborious, expensive, time-consuming, and required by safety level 3, which is expensive to mount. And some of these fungi, I should say histoplasma, is also very difficult to grow on media. So due to the same reasons, as we speak in Malawi, the prevalence of histoplasmosis and aspergillosis is not known, despite the fact that we have HIV prevalence of 11% and the country is classified as a high burden of TB. there's likelihood that cases are being missed and misdiagnosed as TB or sepsis. So the broad objective of the study is to develop and evaluate molecular and serological methods for diagnosis of aspergillosis and histoplasmosis. And then when we go to specific objectives, which is going to be our talk of today, is to evaluate the optimized quantitative real-time PCR method. Just to give you an overview of study design and setting. So this is prospective as well as the retrospective study, because samples are collected from 2 sites. So we retrospectively collect sputum samples from National TB Reference Lab and prospectively from Queen Elizabeth Central Hospital for optimization of real-time quantitative matrix PCR and determination of fungal infection prevalence, respectively. So actually, prospectively, we collect blood and urine specimens from patients presenting to Queen's Hospital who are being tested for fungal antigen and antibody, CD4, and TB-LAM. So there will be also a confirmatory ELISA assays on serum and urine specimens. So I'm trying to give you a project overview in terms of inclusion and specimen, as I indicated in the previous slide, that the enrollment is being done at 2 sites. So under A, these are samples collected from Queen Elizabeth, and we have inclusion criteria, adults above 18 years, samples from TB suspect, whether on treatment or not. The samples we collect sputum, blood, urine, where we are testing serological tests, where we are conducting serological tests and molecular tests. Same as enrollment facility B. However, under facility B, we only collect sputum for molecular testing. So this is a general overview of our real-time quantitative matrix PCR. So we work with 2 primer sets, histo and aspergillus. We have our master mix and the DNA template, everything in one tube, and we use Lotagen to detect pathogens. Just to highlight on critical steps, there were a number of quality indicators, a number of steps involved in developing the real-time quantitative matrix PCR, but just a few of them we started from identifying conserved DNA target, designing primers and probes, determining the annealing and elongation temperature, compatibility testing, specificity testing, low detection limit, determination of CT value, and then initiation of sample testing. So actually, these are the actual primers we are using. We have got 2 sets for histo, but we are using one, much as both sets are working properly, as well as targets, DNA targets. So coming to compatibility testing, we used non-positive DNA samples to be sure if our primers and the target are indeed compatible. So this was done in Uniprex PCR method, where all the primers gave us a positive reaction based on the target. So if you can see like under histoplasma, we have a positive reaction, but there was no positive reaction under fumigators as flavors. So Panaspergillus, this is the mother primer, which is supposed to detect each and every family belonging to Aspergillus. So we registered positive reactions under fumigators as well as flavors. Coming to sensitivity, we registered 100% sensitivity. They were very compatible to the targeted DNA. So we also performed real-time, we also performed the specificity of the method where we use non-positive DNA samples and the non-negative DNA samples. This was done in matrix where we could combine histo and Aspergillus, and we only got positive reactions where it was possible. So we did not get any positive reaction in DNA samples which were negative, which were not suspected to give us any kind of reactions, and our specificity was 100%. We ended up determining low detection limit. So this was achieved by using tenfold dilution factor. Our starting concentration was 1 nanogram. And of course, using the same, we were able to determine number of cycles to work with our PCR. So we knew that after 40, we are not getting anything. We limited our PCR to 40 cycles. To give you the preliminary findings about the prevalence of fungal infections, so far we have tested 233, and we have registered 20% of positive samples, where histo is 30%, and Aspergillus is representing 70%. Now, as I say that we have subfamilies, we have families under Aspergillus, we had to type further and categorize in terms of positivity. We can appreciate that most of the positive cases are for Aspergillus fumigators. Interpretation of the results, just in summary. So we optimize the real-time quantitative multiplex PCR for detection of fungal infection in clinical specimens such as sputum, where we managed to detect the LOD, specificity, and sensitivity. And the initial testing, we have managed to register 20% of positive samples, both histoplasm and Aspergillus fungi in sputum specimen that were collected from the two sites I talked about. So we can appreciate that Aspergillus was highly represented. Future research directions is to finalize molecular diagnostic testing using multiplex PCR for fungal infections, to perform serological assays using rapid antibody antigen and ELISA for Aspergillus and histoplasma fungi, to assess the usability of rapid antibody antigen test at primary and secondary health facility, to determine prevalence of Aspergillus and histoplasma fungi infections in presumed or confirmed TB patients, to develop TB and fungal infection testing algorithm, as we can appreciate that we do get those cases. Allow me to acknowledge Prof. Tony for the wonderful guidance and the support towards the study. Also, Prof. Denise Amsaya, Prof. David Denning, and Dr. Bernard, who are my supervisors. Dr. David Chaima, who is providing guidance in molecular department, Lyson and Joseph Larry, who are providing technical support in the molecular department. Also, not forgetting my funders and those companies which have supported me with reagents and the host institutions. Basically, that is what I had. Thank you so much. Over.
Thanks very much indeed, Pocha. Are there any questions? Nothing's come up on the chat yet. I was wondering, so excellent talk, Pocha, and fascinating findings. So the prevalence of these two fungal infections is really far more widespread than possibly people would have suspected. Do you think then that that makes the case to look at the entire WHO critical list to start with, or do you think you homed in on the two major ones that would be worth studying?
So, pardon, Prof.
Sorry?
May you come again?
Yeah, sure. Are you going to look at the others on the critical list? The WHO critical group. You're muted at the moment, Pocha.
Yeah, so of course maybe connectivity from my end, but at the moment what we understand is that we have this infection secreting in the community, undiagnosed and obviously untreated, because the treatment of histo and aspergillus is different to the usual identified fungi. We normally pay attention to Streptococcus and Candida, so the treatment options are totally different. So we need to dive into like more studies and how we can even accommodate these other fungi and inform the public about the need of including the diagnosis of the two, including other fungi.
Okay. And do you see doubly infected or multiply infected examples?
I don't know why I'm trying to get you clear, Prof.
Are there examples where there is more than one fungal infection in the same sample?
Oh, okay, yes, for sure. So we yet to finalize data analysis, but it's obvious that we are seeing more fungal infections in presumed TB and the TB suspects and the MDR suspect, based on the little data that we have.
Right. And that begs the question, what comes first, the fungal infection, the promoting TB infection, or the other way around? It'd be very interesting to find out. Any more questions? Stefan, you're unmuted again. So,
well, it's thanks also from my side for a nice talk. It's indeed a very important topic. Can you elaborate a little bit about treatment in Malawi in particular? Diagnostics is a problem. So it's underdiagnosed, but it's probably also undertreated. So even in cases where there is a diagnosis, can you perhaps elaborate a little bit?
Thank you for the question. So indeed, we are limited in terms of treatment as far as the two neglected fungi are concerned. So mainly, we are able to diagnose Streptococcus, which is being treated fluconazole and some other drugs. In that area, we are well stocked in the country, but we have challenges with other drugs to support treatment of like histoplasma, which normally we use azole drugs, treatment of aspergillus. So basically, this study will help to inform the policymakers to start like thinking about expanding in terms of stocking drugs, in terms of budget, in terms of reviewing the testing algorithm, etc. But we are indeed limited in terms of treatment as we speak.
Thanks.
Okay, Christine has a question in the chat. So I think you could probably read it rather than me read it out, because you don't hear me so well.
So we have not compared our multiplex PCR results with microscopy. However, for some samples, these will be compared with ELISA as well as antibody antigen testing, much as there might be some kind of differences, because a team from Queen's Lighthouse Clinic is providing sputum, blood, and urine. So we are using sputum for PCR, blood for antibody antigen, urine for antibody antigen as well. So we have not done that.
Lovely. Okay, well, I think in view of the time, we should probably move, but I'd like to thank everybody for attending. We've lost a few people over the time, but that's understandable. Please do come back in a month's time. I think that was a great start. Thank you, Taco. Thank you, Pocha. And thank you, Andrea, for all your organization and overcoming all those technical hurdles for us. Are there any final words you need to say?
Yeah, no, thanks everyone for attending. Thanks for registering, as you also said, Andy. We'll keep updating the schedule and see you in a month, and stay tuned.
Okay. Bye, everyone.